Hi, thanks very much for having me tonight. It's a real pleasure to be here, and it's absolute honor to see that amazing list of speakers that you've organized for the coming months. So thanks so much for being here tonight to share this with me. My talk is entitled From Prehistory to Precision, using the Neanderthal oral microbiome to develop oral microbiome transplantation. Or, if you're a user of ChatGPT, you might translate it into something like Neanderthal oral microbiome extravaganza, pioneering transplants with ancient magic. I think that's the title my students would prefer I use. As I get started, though, even though we're meeting virtually tonight, I would like to acknowledge just the traditional lands in which we sit. Much of this work was done on Indigenous lands, both at the University of Adelaide and now here at Penn State. So I just want to acknowledge elders past, present, and emerging, and just pay gratitude and respect to their ancestral spirits. I also want to mention that there are some human remains in this talk. So if that is an issue for you, please take care as we get towards the middle of the presentation tonight. So I want everyone to think for 1 second just about what the most widespread chronic disease is in the United States. Or if you're internationally, you could even think about this in your own country. What's the most widespread chronic disease? I think it might actually surprise you, because the most chronic disease in the world today, or at least in the United States, is the presence of dental decay or dental cavities. In fact, 90% of people in the United States will experience a dental cavity at least one time in their lifetime. 90%. That makes this the most widely experienced chronic disease in the United States today. And in fact, other oral diseases aren't far behind dental decay. In fact, 70% of Americans over the age of 65 will also develop periodontal disease at some stage in their life. 70%. These are absolutely enormous numbers. And we treat both of these diseases with a technology that was actually developed in 1938. In fact, this is Dr. West's Miracle Tough toothbrush. I hope you can appreciate that that toothbrush from 1938 doesn't look all that different from the toothbrush that we have today. But what this effectively means is that we're using an 80-year-old technology to treat some of the most chronic diseases on the planet today. And in fact, in the United States, 50% of Americans still use a toothbrush that looks very similar to that version that came out in 1938. So really, we're treating the most chronic disease in the United States with an 80-year-old technology. I think we might be able to do better. And in fact, much of the expertise for why we should treat these diseases this way actually also stems from the sort of 1940s. In fact, post-World War II, this was the era where we really started thinking about taking care of our oral health and brushing our teeth. And in fact, some of this actually stems from the military. This is a screenshot from one of the sort of training videos that was given to new recruits during World War II. We were really taught to keep, they were taught to keep their oral health in great condition, as that was one of the reasons for sort of winning the war in that context. But I hope behind the person on this screen, you'll see number one there, keep your teeth clean. That was the most important thing. And this really stemmed from an antimicrobial era. This is about the same time that we developed antibiotics, and they became sort of mainstream in our populations today. So I really hope that you can appreciate that this sort of, again, technology developed in 1938 really is working to kill microbes and keep them, really at bay. But again, this is sort of an 80-year-old idea, and it really hasn't gotten us anywhere if these are the most chronic diseases we have today. We really need to do better, and we need to create better preventative treatments for oral disease. That's the bottom line. And I would argue that we should actually do this by not thinking about this in an antimicrobial perspective, but actually by thinking about this in a pro-microbial perspective. That's because we live in a microbial world. Every surface we touch, every sip of water we take, every ounce of air we breathe is littered and full of microorganisms. And the human body is no different. In fact, we have over trillions of bacteria, fungi, and viruses that we lovingly call microbiota that live within the human body. Now, most estimates suggest that this is more than 50% of the total cells in the body, and that there are over 1,000 different bacterial species present between your gut and your mouth and your skin and all of the different body sites that you have. 1,000. Try thinking about 1,000 different mammals for a second. It's pretty difficult. And that equates to about 1.4 kilograms or 3 pounds of your total body weight. That's analogous to your brain. So you essentially have an extra organ's worth of weight that you carry around with you all day, every day. Now, most of this audience, I probably don't need to explain that to, but I hope if you're new to this, that provides a little bit of light as to how important these microbial communities can be for the body. Because it's not just their presence that's really critical, it's also the functions that they carry with them inside of their genomes. We refer to that as the microbiome. Now, best estimates are somewhere between 2 to 5 million bacterial genes within the human body, or sorry, within the within the microbes within the human body. And if we're being really generous and we assumed that there was maybe 30,000 human genes, that means that 99% of your genetic makeup is actually attributable to microbes. Now, if we're trying to think about how we cure diseases or how we create new preventative ones, we are certainly going to have to think about this other 99% of the DNA present within the human body. These microbial communities and the functions they bring with them are also really critical causes of disease. Now, in the mouth, we know that the oral microbiome is directly and indirectly linked to both oral and systemic diseases. The diseases I mentioned earlier, things like caries and periodontal disease, are indeed sort of linked to directly causing, I should say, the sort of diseases in the mouth, whereas things like oral cancers can be more indirectly linked to the oral microbiome. But we also know that systemic diseases, things like metabolic diseases, mental health, and even gastrointestinal cancers and cardiovascular disease can all be both directly and indirectly tied to the microbes living in the mouth. So this makes it, really, really important that we think about these new preventative treatments in a way that leverages microbial ecology. This really provides a new path forward to think about how we might shift microbes and prevent disease. Now, in my lab, we actually take a very unique approach to this question. In fact, we use anthropology and archaeology to do so. And I think anthropology and archaeology both have a really key role to play in understanding how we cure new diseases. And that's primarily because we can trace back the origins of these diseases using ancient DNA and using the past. So really, archaeology has a key role to play to tell us how we got the diseases, why we got the diseases that we have today. In anthropology, studying these across diverse populations might also shed light onto the very specific mechanisms that cause disease, regardless of your human genetic background or makeup, or regardless of your environment. So it's really important to study diseases broadly and understand them both through time and across space. And tonight, I'm going to tell you about 3 different areas of research in my lab that sort of play a role in trying to come up with new treatments today. I'm going to tell you a little bit about the ancient DNA work that we've done in Neanderthals and other anatomically modern humans, and how that really fuels our search for a really healthy oral microbiome. I'm going to tell you a little bit about oral microbiome transplantation and how my team has been working to develop that technology. And last but not least, I'll tell you about how we're trying to develop that technology in an equitable way, using studies across different cultures. So let's go ahead and get started. One of the ways we use sort of a time machine to go back into time is by using ancient DNA to reconstruct where these diseases have come from and how we have the microbes that we have today. Now, ancient DNA can be obtained from a wide range of different specimens. Perhaps you've heard of studies about the Neanderthal genome that came from ancient bone, or maybe you've heard about other studies in soil, feathers, hair, or even eggshells. But ancient DNA is classically very difficult to work with, and it doesn't survive forever. In fact, some of the best estimates for the half-life of DNA suggest that it might not be recoverable for more than 1 million to 1.5 million years. In fact, the oldest ancient microbiome study that we would consider valid or authenticated is no more than 2 million years old. So unfortunately, Jurassic Park is not real. I'm sorry to burst everybody's bubbles. And it does mean that we also can't get DNA out of things where DNA would not survive well, where it's been destroyed or chemically treated, and things like amber and Egyptian mummies. And even if you're able to recover ancient DNA from specimens, it's incredibly frustrating to work with. And in fact, ancient DNA is subject to DNA fragmentation. Our DNA breaks down to incredibly tiny pieces. In fact, the average DNA length of DNA sequences from our Neanderthals was actually only about 38 base pairs. We also know the DNA is damaged. We actually get the wrong DNA sequences out of some of our ancient specimens. But we can also use that damage to authenticate much of our ancient DNA from modern DNA sequences. We know that there's taphonomy in ancient DNA. We know microbes with different cell walls and different cellular structures might degrade at different rates, and that can really change how we interpret our results. And last but not least, we know that things might be highly contaminated. Many of the individuals that we work with have been buried in the soil, and we would expect soil contamination to be part of our equation when we're trying to reconstruct authentic and robust microbiomes from the past. So my team has been working on many of these problems over the last 10 years, really trying to remove bias and authenticate and make sure our signatures are as robust as possible as we move forward. So using a variety of these new tools, we've been reconstructing the oral microbiome from ancient populations from somewhere about 50,000 years to the present. And we do this using a very unique sample type called dental calculus, which is just calcified dental plaque. Now, if you've ever had a sugary drink and you might feel that slime forming over the surface of your teeth, that is a bacterial biofilm growing and reproducing in real time. And these microbes, when you sleep at night and the pH in your mouth changes, calcify into a rock-hard-like matrix. And that continues to grow each night as you sort of go to sleep, unless you're somebody who goes to the dentist and has it removed through scaling or planing or even sonication. The really awesome part about dental calculus is that it survives in the archaeological record. It survives as well as bone, and we know it dates back into the archaeological record for millions of years. And this really provides an easy way for us to access oral microbial communities of the past and understand how and why we got the types of microorganisms that we have today that are now associated with disease. So one of the first studies we did to look at this was actually to go back into other hominin species that have great oral health. And in fact, many wild chimpanzee populations, as well as many Neanderthals, have excellent oral health. We wanted to look at anatomically modern humans from prior to sort of farming, but honestly, those individuals can be difficult to identify in the archaeological record. So Neanderthals can almost serve as a proxy for many of these folks in the past. And then we compared these to modern humans that lived in Adelaide, Australia. And one of our first findings ended up being one of the most important. Now, this is a really simple sort of pie chart showing the different bacterial phyla and archaeophyla that we obtained from dental calculus from chimpanzees, Neanderthals, and anatomically modern humans. And what I hope you can appreciate is that there's many more blue pieces to the pie in the chimpanzees and Neanderthals versus the red pieces of the pie in the modern humans. Those blue sort of pieces of the pie are gram-positive microorganisms. Many of these are associated with oral health, and they're thought to be things that we'd want to have in our mouth. The gram-negative organisms, many of these can stimulate the immune system and are in fact associated with things like periodontal disease. So straight away, we see that there's a bias towards disease-causing microbes in modern humans. We also see that Neanderthals have archaea present in their mouths, which are largely missing in modern humans today. We then compared these to a wide range of anatomically modern humans through time to really, again, track back when the sort of shift to these disease-associated microbiomes started occurring. So we compared these Neanderthals and chimpanzees to anatomically modern humans living in both Africa and Europe. And in fact, the first change that we saw was slight in the oral microbiome, but it was indeed when people started hunting most of their food. And in fact, when we move from foraging to hunting, we see that sort of slight change in the microbiome. But the largest change, and the one that initially is associated with disease, actually happens in Europeans when they start practicing agriculture, probably about 7,500 years ago. We actually start to see more of those red bars that correspond directly to the plot I was showing you earlier, and many more of these sort of gram-negative organisms coming into the mouth. We also see a significant rise in fusobacteria, which can also help facilitate the building of large calculus fragments or large plaque in the mouth. So really, this shows us that we've had, a major transition or several major transitions, but really that the adoption of agriculture is something that should be associated with the sort of decline of oral health, at least in European populations. But we wanted to dig into that a little bit further. What happened after folks adopted agriculture in Europe? And why did that all of a sudden become associated disease as we got closer to the modern era? One of the things we did was zoom in on a particular location in order to control for geography and control for any other confounding effects that we might see. And in fact, we just published this study a couple months ago, and it was led by Abby Gans, who's in my lab at Penn State, and Andrew Farr, who's now at the Cambridge Science Center. And in this study, we looked at 235 individuals or ancestors who lived in Great Britain. This included 127 well-contextualized adults from London. And this is really important because these folks from London have been incredibly well studied, and we have really great information to contextualize their cultural, religious, environmental sort of areas that they lived in, all the information that we would sort of want to look at, as well as their health status. It makes them a very valuable data set to examine through time. So one of the things that we saw straight away when we looked at the oral microbiome of folks in Great Britain was really the variation being split by two distinct microbial ecologies. And so in this PCOA plot, where every single dot represents an oral microbiome from an ancient individual, we really see that it's really dominated by the presence of two distinct ecologies on either end, and then sort of a gradient in the middle. The ecology shown there in green on the right is dominated by species in the genus Streptococcus, whereas the one on the left is dominated by microbes within an archaea genus called Methanobrevibacter. And so these two distinct communities were really shocking to us. In fact, we were really quite shocked to know that the Streptococcus community is also what associates to what most folks have today who are of European descent and live in industrialized communities. Whereas the Methanobrevibacter community on the left is something that hadn't really been fully described in living populations. Some of these Methanobrevibacter species have been reported in modern populations, especially those with periodontal disease, but yet we hadn't really described entire whole microbial communities associated with these. And in fact, in network analysis shown there on the bottom right, we do see that these microbial communities are really led by entire different ecologies. It's not just dominant taxa that are different, it's the entire microbial community that's really shifted or that is different in these individuals. And so we then sought out to find how we then got the Streptococcus community to be more dominant in current populations and what could have driven whether or not people have either one of these two microbial communities. And honestly, we tested many factors that we hypothesized from both the ancient DNA literature as well as the modern oral microbiome literature as to what would have been driving these sort of differences. And in fact, the only thing that we saw to drive this difference was actually an association with time. And when you break this sort of effect down into different periods and different blocks, what you see is the shift of this microbial community in the past is actually linked to the arrival of the second plague pandemic in London. And so you might have heard of this as the Black Death or Yersinia pestis, but this was when 30 to 60% of the population passed away in a matter of a few months. So an absolutely devastating pandemic. And while we could have things that selected, well, we could be seeing, an effect where the microbial communities that survived were just the effect of people who actually survived the pandemic, there were a lot of social and cultural factors that also changed after this time period. In fact, there were fewer workers. And so workers who were around could ask for higher wages and ask for better diets. And in fact, there's quite a bit of isotopic evidence to suggest that people who survived the pandemic were able to access higher status dietary items after this period. And so really thinking about more meat in their diet. So we really wanted to test some of those effects on the types of microbiome, in addition to, potentially direct hypotheses. One of the things that we did was actually look for functions associated with different dietary types present in the microbiome. And lo and behold, we actually found that dietary functions associated with things like meat consumption, dairy consumption, and sugar consumption were all present in the Streptococcus microbial communities that Europeans from industrial communities now share today. And this was opposite from what we found in folks who had the Methanobrevibacter-dominated communities. And so this suggests that while there may be direct effects, indirect effects like culture and society and the diets that people were eating may have also helped select for the types of microbes that we have today in our mouths, the Streptococcus-based community. So we thought, gosh, maybe this is really great. Maybe this is telling us something important. If we think the Streptococcus community is what's associated with disease today, maybe we should be looking at this Methanobrevibacter community. Maybe that's what everybody should have in order to support their health. And so we use these really well-contextualized individuals from London to understand whether or not these microbial communities were associated with a health status. We indeed find that the Streptococcus community is associated with things like periodontal disease, but unfortunately, we also find that the Methanobrevibacter community is also associated with signs of systemic disease, things associated with systemic inflammation, such as periosteitis, osteophytic lipping, and porosity of the bone. So this suggests to us that, really being able to try to select for individual microbial communities might be much more difficult than we initially thought. And that just because microbial communities aren't present today does not mean that they're necessarily associated with health. We've also gone on to do a similar study in the Americas, predominantly on the west coast of South America, in collaboration with folks in Azapa Valley, as well as in Miraflores. And one of the things that we see is that this story is also going to be completely different in other parts of the world than it was just in the UK. So although we're able to tell the story and the history of how we got the microbes we have in the UK, we know that different types of microbes exist elsewhere in the world. And in fact, on this particular tree, each of the green dots represents folks who lived in the Americas at the same time period in the past as all the blue dots, which represent most of the British paper I just talked about. And in fact, we see that unique microbial communities dominate folks that lived in the Americas. So this suggests to us that we're going to need to reconstruct evolutionary histories and their association with disease uniquely in every single one of the populations that we want to study and that we want to understand. So I'll close out this first part of the talk and just suggest that Neanderthals, primates, and even anatomically modern humans living before the adoption of agriculture likely had a healthy oral microbiome, something that's very different than what people had today. We also know that things like social cultural transitions, including things like diet, may have helped select for the types of microbes that are now associated with our most common chronic diseases. And we also know that the evolutionary history of our oral microbiomes may be different across many of these different populations, which means we have a lot of work ahead of us. But what could we do if we were going to take this ancient DNA data and actually translate it into the modern day? How would we go about doing that? Well, there's a variety of potential methods. I'm sure some of them you've heard of, things like probiotics or prebiotics, either giving both specific individual species of microbes or giving prebiotics, things like healthy foods to microbes, so they might want to grow and do better. You can also think about phage therapy. Maybe we should try to eliminate the types of microbes that cause disease in the mouth. Or, last but not least, you could think about a transplant. This has been predominantly pioneered using fecal microbiome transplants, so the ability to take microbes from someone's feces and transplant that into a recipient to improve health. And in fact, there are many systems and processes that now have been developed to help support that. And I think my favorite might be called the repopulator, shown there, where you can actually grow fecal microbial communities up as a way to standardize them before transplant. But things like microbial transplants simply don't exist yet for the mouth. They haven't yet been developed. And so I worked with a team of researchers at the University of Adelaide that were led by Lisa Jameson and Peter Ziln to really work together to develop oral microbiome transplantation, or what we call OMT. And a lot of the work that I'll show was led by PhD students Sonia Nath and Kevin Ketagoda on the team, who've worked incredibly hard over the last 4 years to get this technology off the ground. So the short of this technology is that we really are trying to, again, use the past and use information about what a healthy microbiome might look like to use that information as fodder to transplant healthy microbiomes into people who might need them. And so we would go through, the idea was that we would go through and we would select ultra healthy donors. Now, these might be people who've never been to the dentist, they've never brushed their teeth, and they've never had any oral health issues in their entire life. And we would collect their dental plaque and we would grow it up in an in vitro flow cell shown there in the middle. Now, in that flow cell, we would be able to grow plaque to large quantities. We would be able to screen it and to probe it and really understand what drives differentiation of healthy oral microbes. And then we could put that into a gel and actually transplant that into a recipient who might need it. You could think of this as being a gel that you sort of squirt in a tray and then put on your teeth, much like you would do with a whitening kit. So we set to work sort of developing this technology. And at first, we did find 100 ultra-healthy donors to donate their oral microbiomes in for this cause, which is a pretty gross thing when you think about it, to sort of donate your dental plaque for science. But I'm so glad that they did. And in our donors, we essentially see many different factors that drive variation across them. In fact, there was no single factor that sort of designated whether or not somebody would be considered ultra oral healthy or not. There were many different features. We did see a large split in our data depending on how often someone brushed their teeth and sort of flossed. You can see those parameters there. But there were many other factors that also sort of guided this. And so, you know, folks who might have thought, oh, well, it's just going to be the people who have really healthy diets and don't eat large amounts of sugar. Well, that's not necessarily true. Some people ate up to 300 grams of sugar and still had, on a daily basis, and still had really great oral health. So this really suggested to us that we were studying unique populations that aren't typically included, I think, in many of the studies examined today, and really lets us know that we also have a long way to go before we understand ultra-healthy mouths. But we took these donors, the 100 different people who donated their dental plaque, and we grew them up in these in vitro flow cells. And you can see the flow cell sitting there on a black heat map where there's a little insert on the right-hand side. And essentially what we do is push artificial saliva medium across this flow cell and we inject the donor's dental plaque into there. And each one of those white circles or white discs is hydroxyapatite, which is like human enamel. And so we can grow dental plaque on those sort of enamel-like discs for up to 21 days. Our preferred time is actually 10 days. And then we can open up the flow cell, remove those discs, and take the dental plaque for transplantation, for genetic sequencing, for metabolomics, microscopy, you name it. And that's exactly what we've gone ahead and done. In fact, our microscopy shows that every single donor has a unique set of microorganisms just by visually looking at them, and that their biofilms are assembled in many different ways. But the good news is that we also have lots of live cells. In fact, over 70% of the cells in our biofilms are still alive after 10 days of growth. We do know, though, that trying to grow these oral microbial communities and flow cells does result in a decrease in diversity, albeit it's a minor one. In fact, we can still grow at least 130 species from any of the individuals that we've looked at so far. And in fact, we only have about a 10% reduction in alpha diversity when we go through that process. One of the surprising things to us was actually about how much individuality of donors is maintained after they go through this flow cell process. And in fact, here you can see the collected or inoculated plaque from individuals in circles and squares on the left-hand side. And you can see that each of those donors, shown in a different color, actually maintain their sort of individual signatures even after 14 days of growth in the flow cell. We then took these donated plaque that had been grown in these flow cells, and we've looked at their ability to prevent caries in a rat model. Here we used female Sprague-Dawley rats, and in fact, we fed all of them a cariogenic diet, but in some of them, we also infected them with Streptococcus mutans, which is a microbe known to cause caries, at least in humans. And in half of those mice, we then gave them an oral microbiome transplantation. In fact, we put that plaque in a hydrogel and we painted it on the surface of anesthetized rats once a day for 3 days, and then tracked them over the course of 5 weeks. And during that particular study, we looked at both the ability to prevent caries as well as safety to try to see whether or not this could be a good idea to do in humans. And this is sort of preliminary data coming out of this study. But one of the things that we see in mice, regardless of whether or not they were given Streptococcus mutans, is that oral microbiome transplantation is significantly effective at preventing dental caries. And this is a really big deal. Most technologies don't sort of have this effect. And we're really excited about how we can think about microbial sort of approaches and transplantation as a new method to prevent dental caries, both in rats and in humans. So we're really excited to just conclude this OMT part of the talk about this new technology and its ability to prevent caries. And in fact, we're sort of working on periodontal disease models as we speak in mice. And so I'm really excited to see that data. And that the ability to grow these sort of dental plaque biofilms in an in vitro flow cell provides a lot of really unique opportunities to test and examine donor plaque and understand who might be the best donor for which particular situations. But with any good experiment, in fact, many more questions remain with our OMT technology. We're still going through safety data, so we really want to know whether or not it's safe. We're really interested in whether or not OMTs will permanently alter oral microbiomes, and including their evolutionary history and trajectory. And if it does, what sort of health consequences may that have for somebody? Especially if you're doing this in children. Does it mean that they have sort of a shifted or altered oral microbiome for the rest of their life? We're also really interested in how we make oral microbiome transplantation accessible, affordable, and equitable. And on top of that, we want to make sure that it's equally effective in all people, right? If we have a technology that everyone can access, but it only works half as well in some people as it does in others, then I don't think we would have done our job correctly. And so the last part of the talk is really focused on this last question. And it's really our work using anthropology to understand whether or not OMT might be effective in all people. And a lot of this work stems from sort of a global comparison that's been done by Iunoloa, who's in my team here at Penn State, and Muesli, who is in my team at the University of Adelaide, where we've been comparing oral microbiomes from our group, as well as that published from other teams, across the world to understand whether or not the same types of oral microbiomes are associated with health. And here is a PCOA plot of these many different studies doing sort of a meta-analysis of salivary oral microbiomes that exist today. And I will tell you that we've done a lot to try to control for contaminant effects and kit effects and all sorts of issues. But one of the things you can see that's the same in the mouth as it is in the gut is actually the fact that industrial oral microbiota is sort of the single biggest factor that drives oral microbiome variation on the globe today. So it doesn't matter if you're living in sort of Africa or South America. If you're living a non-industrialized type diet or lifestyle, you're going to have a different oral microbiome than folks living in sort of quote unquote industrial communities. Now, I think the labeling on this is incredibly problematic, but we've stuck with the labeling that's currently used in the microbiome field, but I'm happy to talk about that in the questions later tonight. We also wanted to understand whether or not the microbiomes in people who are not living in, or not from European descent or not from industrial communities are associated with disease in the same way, right? If we're going to try to target ways to improve oral health, we have to know the multitude of different ways that the microbiome might cause disease. And so here we've worked again with Lisa Jameson, and this was work led predominantly by Matilda Hanley Davis, who was in my team at the University of Adelaide, and is now a science writer in Copenhagen. But one of the things we did was work with Aboriginal Australians and Torres Strait Islanders to understand their microbiomes and how they associate with periodontal disease. And in fact, we do see that Indigenous Australians have a much higher diversity of microbes compared to non-Indigenous Australians. And in fact, there are actually 127 different species that were present in Indigenous Australians that had not yet been described in the HOMD, or the Human Oral Microbiome Database, the current gold standard for describing the types of bacteria that live in the mouth. We then went on to compare Aboriginal Australians and non-Indigenous or European descent Australians living in the same city who were all suffering from periodontal disease. And in fact, even folks who are suffering from periodontal disease still sort of separate by culture there. You can see Aboriginal Australians shown there in the green dots. So again, regardless of the disease state that's described the same, we see different microbial communities underpinning those diseases. And so again, it suggests that we're going to really need to think about the types of microbial communities we're using to cure disease and make sure that they're not harmful in another population. I think of interest to me is that there are many microbes in Aboriginal Australians that we have no idea what they're doing in the human body. One of the microbes we find in Aboriginal Australians living in the central desert is actually tied to the genus of microbes called Endomicrobium. Now, Endomicrobium you may recognize as being a microbe from termite guts. It's the microbe that helps digest cellulose and allows termites to get energy from food. In fact, central desert Aboriginal Australians ate termites for tens of thousands of years as an excellent source of protein. Now, these folks are living in cities, specifically in Adelaide and Alice Springs, and so we know they're not currently still eating termites, but their sort of heritage and that evolutionary history may play a big role in the types of microbes people have today, as well as how those microbes are linking up to disease. And so we really have to be aware of how these diverse microbial communities cause disease in all different people to really make a technology and to make OMT equitable and effective across all populations. We're working with a wide range of other Indigenous communities. We're working with the Turkana Health and Genomes Project in Kenya, with First Nation folks out of Manitoba in Canada, with the Ghana, continuing a very large project there in Adelaide, South Australia, and with the Shivanche in central Brazil, to really, again, understand how diverse the oral microbiome might be and how these microbial communities are associated with disease. Because if they're a uniquely associated disease, we need to know about it so that we can create technologies that don't do more harm than they do good. So our anthropological work has really shown that industrialized oral microbiota are probably unlikely to result in effective disease prevention in all people, simply because we share very different microbiomes depending on where we live in the world. And so we need to consider genetic, cultural, and historic diversity as we're developing this technology. And in fact, personalized approaches that really leverage one's culture, heritage, or even their identity might really be the key to equitable access for this technology. So tonight, I hope I've been able to keep your attention, but I've really talked about how leveraging microbial ecology really may open new doors for oral health therapies. I hope I've shown you that archaeology and anthropology can play a really critical role in how we develop OMT technology and how we shape our future oral health. I also hope you've taken away that equitable, ethical approaches to the development, commercialization, and distribution of OMT technology are really critical to making a dent in how badly caries and periodontal disease influences our health today. So with that, thank you so much for listening. I'm eager to hear all of your questions. And yeah, thank you so much. Follow us on social media.